So guys, let's we finish our discussion on the carbohydrate metabolism in plants. Today we will talk about a very important carbohydrate which is present in plants. It's one of the most abundant carbohydrate also. It's cellulose. It's a fibrous carbohydrate. Specifically, or most of the time, it is found in the plant cell wall. It's, as I've already told you, it's one of the most abundant carbohydrate on earth. 50% of all the carbon which is present in the plants is contributed by cellulose. Cotton fibers and paper are about 95% of cellulose. It shows you not only the importance of cellulose, but also that it is how it is industrially or commercially significant compound. Similarly, 50% of the wood also contains cellulose. Again, it shows the commercial importance of this compound. Similarly, the dry weight of the plant leaves is around 10 to 20% made up of cellulose. As far as the structure of the cellulose is concerned, it's not something very dissimilar from the other polysaccharides you have been studying. Like for instance, in animals it's glycogen, and in plants another very important polysaccharide is the starch. Again, it consists of glucose monomers which are linked with each other through glycosidic linkage. The glycosidic linkage is of beta-1,4 type. So this is something different from the glycogen or the starch, where you saw it was alpha-1,4 linkage. Also, another difference between the plants, the cellulose and the starch and the glycogen is that in plants, the cellulose is exclusive, actually totally, it's a linear shape. I mean, there is no branching in the structure of the cellulose molecule in plants, cellulose molecule, but in starch and glycogen, there is branching also. That is because of the alpha-1,6 glycosidic linkage. So the cellulose molecule is composed of linear arrays of the glucose units which are linked through each other through the beta-1,4 linkage. And also it's this, because of this linear structure and because of the very composition of it, it allows very extensive hydrogen bonding between the hydroxyl groups which are present in these sugar moieties. Another very important thing about cellulose is that it's very inert. It doesn't do much interaction with water or other solvents, which contribute towards the stability of plant. So it gives you know the rigidity in the wood. Similarly, the rigidity provided by the cell wall, it's because of this very nature of the or this feature of the cellulose, that it doesn't get dissolved into water that much, it doesn't interact that much with other solvents. As far as the synthesis of cellulose is concerned, it is made up by the continuous addition of the glucose monomers, and the glucose monomers are added in a way that the glucose is first connected with or bounded with the GDP or UDP, and you know this, that why we bound the glucose with these molecules, because it make glucose more energetic so that the reaction will be feasible. So whether the intermediate will be GDP or UDP, it will depend upon what sort of synthase is used, that is what sort of cellulose synthase is being used. If you have been following my previous lecture, so it must struck you that there is some similarity in terms of formation of the compound, the cellulose compound. There is some similarity with the other compounds also you have been or we have been discussing. For instance, if you remember the glycogen synthesis, so this figure, if you see it, you can see that the synthesis of cellulose is very similar to the synthesis of glycogen. Glycogen में क्या होता था? That glucose 6-phosphate was converted into glucose 1-phosphate, and glucose 1-phosphate was then bonded with the UDP because of the with the assistance of UTP, so that you eventually got the UTP glucose molecule, which then was added up into the growing chain of glycogen. Similar way, you do the same, you use the same glucose unit, same utilization of the glucose occurs. The only difference is that here in sometimes not only UTP but also GDP can be bounded with glucose. And again, I have told you that it depends upon what sort of cellulose synthase is being used. And also another difference is that, of course, because the bonding is occurring in a way that it is not alpha 1,4 linkage, but it's beta 1,4 linkage. So otherwise it's very similar. Then not only that, you have now arrays of molecules, cellulose molecules you can say, which are having small units of the glucose moieties which are arranged in an array, but also then they are aggregated, reorganized in the form of small fibrils known as microfibrils. These microfibrils are further reorganized to form larger structures known as the cellulose fibers. You know, I mean, if you see the cell wall, it has a very organized structure, and this organization occurs because of this organization which is formed during the synthesis of the cellulose molecules. First you have the individual arrays of the cellulose molecule consisting of units of glucose molecules, and then these arrays are aggregated in the form of microfibrils, and these microfibrils further rearranged to give rise to a very meshwork of structures known as what we say the cellulose fibers.